Good morning, everyone. I am Assistant Professor J.C. Panigrahi. Welcome you all to this second slot of AICT-sponsored Sttp on integrating research in basic science with applied pharmaceutical science, organized by Roland Institute of Pharmaceutical Science. Let's begin the session with a virtual lamp lighting.
कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः
Now I would like to request our respected principal sir to
वक्रतुण्ड महाकाय सूर्यकोटि समप्रभा निर्विघ्नं
Good morning, everybody. It is always a pleasure and privilege to welcome the meritorious and bright student of Roland Institute of Pharmaceutical Sciences, Dr. Rabinarayan Parhi. I welcome you, sir, to this short-term training program sponsored by AICTE.
Thank you, sir.
And I hope your lecture will definitely benefit all the delegates. I request Dr. Gautam Kumar Jana to introduce today's speaker, Dr. Rabinarayan Parhi, to the delegates.
Thank you, sir. Good morning, one and all. I, Dr. Gautam Kumar Jana, Associate Professor, Roland Institute of Pharmaceutical Sciences, would like to introduce the speaker of today's session, Dr. Rabinarayan Parhi. Dr. Rabinarayan Parhi is currently working as an Assistant Professor in the Department of Pharmaceutical Sciences, Susruta School of Medical and Paramedical Sciences, Assam University, a central university, Silchar, Assam. He had started his journey in pharmacy from Roland Institute of Pharmaceutical Sciences, Berhampur, in the year 2001. He had his post-graduation from the Indian Institute of Technology, Banaras Hindu University, IIT, BHU, Varanasi, in the year 2008. With pharmaceutics as specialization, he obtained his PhD from Berhampur University, Odisha, in the year 2017. He has published papers in high-impact journals like Material Science and Engineering C, Journal of Advanced Research, Environmental Chemistry Letters, Drug Delivery and Translational Research, and Journal of Drug Delivery Science and Research. He has more than 50 publications in reputed international journals. He authored 4 books and published many book chapters in Elsevier, CRC, and Springer Nature publishers. He is serving as an editorial board member as well as reviewers in many reputed journals under publishers like Elsevier, Springer Nature, Taylor and Francis, Wiley, and Bentham Science, etc. He is PI of UGC-funded research projects. He has delivered many invited talks in universities and institutes in India. He has research experience in transdermal and intradermal drug delivery, penetration enhancers, microneedle technique, and 3D printing of pharmaceuticals. Dr. Parhi received a research award in 2010 from IDMA from his research on natural penetration enhancers. He is a life member of APTI, IHPA, and IP GA. With this brief introduction, I would like to welcome Dr. Padi to deliver his lectures.
Okay. So good morning to all. Thank you, Gautam sir, for introducing me beautifully. At the outset, I would like to thank the entire management of REEFs and particularly Principal sir, Ravi Kumar sir, Vice Principal sir, Niranjan sir, and coordinator of today's program, Gautam sir and Kanu sir. And let me share the slides.
I think you can directly open your PowerPoint that is there now at the bottom.
Yes. Yes, sir.
You
It's
open
coming
that.
now, sir. Yes.
Yeah, it is coming.
Yeah. Thank you.
Make it full.
Yeah. So now today my topic for the is micro needles, a third generation transdermal drug delivery technique. So basically, why I have chosen this particular topic is because I generally worked in the transdermal and interdermal your delivery systems so far, and the microneedle is today innovative and most successful physical technique to increase the penetration of drugs into the skin as well as into the systemic circulation. So with this, I'll move to the topic introduction part. So here, basically, when we are talking about any topical preparations, we are going to apply on the skin. We have two targets. First target is skin. That is when the target is skin, that will be called as dermal drug delivery. And if the target is your systemic circulation, that will be called as transdermal drug delivery. And when we talk about the generation in transdermal drug delivery, we are having generally 3 generations. Why we're talking about the generation? Because since the inception of transdermal drug delivery, we are having many techniques to improve the permeation of drug across the skin. And we know that in 1979, first your transdermal system, transderm scope, is came into the market with the approval from FDA. We have seen many formulations, transdermal patches came into the market. But there are certain problems like the limitations of drug permeation into the systemic circulation by the epidermal layer, more particularly I'll say stratum corneum, will prevent generally the drug permeation into the systemic circulation. Then many techniques came into the picture, and those techniques are divided into generations where the mechanism of action and the way of drug delivery is considered. So the first generation is talk about the diffusion of drug into the systemic circulation. Second generation is talk about non-invasive drug delivery with certain cases the actuation or external stimuli. Third generation is minimum invasive transdermal systems. In the minimal, sometimes we are calling it as non-invasive also. The microneedle technique is also called as non-invasive. Sometimes we are calling minimum invasive. Depends on to what extent we are going to penetrate the microneedles into the skin. So the first generation is talk about the passive diffusion of the drug into the systemic circulation, where the traditional patches will come into the picture, like membrane permeated, reservoir system, matrix adhesive systems, where the main problem associated with the physicochemical properties of the drug. It is certain that the drug molecule below 500 Dalton will only be able to penetrate into the skin, and the daily dose of the drug should be less than 10 mc per day. So, and also we should have the ideal log p value between 1 to 3.5, and aqueous solubility also requirement. So with all these requirements, only few drug molecules are qualifying to be included in the transdermal systems. So then the second generation came into the picture, where we can enhance the drug permeation with the external stimuli, and also the particulate systems will come into the picture, like emulsions and nanocarriers. So here the large molecules like peptides, proteins, vaccines, DNA molecules are unable to penetrate into the skin. Even if we are having the external stimuli like magnetic field or electrically assisted techniques like iontophoresis. So then we came into the third generation, where invasion, or we can say minimum invasion, is the consideration, where we will be having cavitational ultrasound, where the mechanism will be cavitation, electrophoresis, microdermabrasion, thermal ablation, and microneedles. So microneedles, among all, the microneedle technique is very successful because of patient compliance, and also it will improve the bioavailability of the drug. So the next slide here, we look at into the development history part, how this microneedle technique came into today's today's world. So now the first microneedle, the word microneedle, came into the picture in 1921, where the microneedle was used for the dissection of animals or insects, we can say. So Robert Chamber used first small needles to dissect the insects, but it is not used that time for the drug delivery. But in 1971, the first concept of MN, that is, we can say solid and hollow microneedles, came into the picture, where the microneedle was patented and the approval got in 1975. Then again, there is a gap in bringing the microneedles to the market or clinical part, where in 1998, we have seen the first research report came to the readers or researchers, where the drug calcin is used for the drug delivery into the systemic circulation. So then in 2001, first report on drug delivery of genetic materials was observed. In that case, Alja corporations bring out this delivery of genetic material into the systemic circulation. Then we come into the 2002, where we've seen vaccine delivery. Very clear. So vaccine delivery is one of the major area for the microneedles. We'll discuss all those later on, later slides. In 2003, we have seen first research report on delivery of macromolecules and nanoparticles with the help of microneedles. Then the progress, it progresses like anything, and we got three things in 2005. One is the first report of dissolving microneedle. This is the advancement or progress in the microneedle, in the designing part. Then we have seen the diagnostic use of microneedles for skin diseases like acne and dermatitis, all those. And 2005, we've seen the cosmetic applications, right? The cosmetic applications. So now we look at what are microneedles. We talk about microneedles and how can you define microneedles? These are micron-sized needles employed painlessly to create transient aqueous conduits of micron size and dimension across the skin that enables delivery of small drugs as well as large drug molecules. So in the diagram, we're seeing the dimensions of the microneedles. So then the microneedle device, we are generally talking about other words related to microneedles are microneedle arrays, where the number of microneedles will be arranged properly on a transdermal patch, normal transdermal patch. Then we look at into mechanism, how they are acting, how they are able to deliver the drug into the systemic circulation. So it generally causes disruption in the skin, more particularly in the epidermis, and thus bypassing the barrier layer of the epidermis, that is stratum corneum, which is made up of corneocytes, dead cells, embedded in bilipid layer, which is the main barrier for entry of any foreign materials or drug molecules into your bloodstream. Then this led to the drug delivery directly to the epidermis or apodermis. Then it will move into the systemic circulation when it reaches to the blood capillaries. So here main aspect is we will not have pain. Non-invasive, or we can say minimum invasive, here we will not get the pain compared to the hypodermic needle, where the hypodermic needle is inserted to a depth where it reaches the subcutaneous layer and muscular layer. So obviously it will touch the pain receptors present in the dermal layer. So we are getting pain. But in case of microneedles, we're delivering the drug in the upper layer of the epidermis, or lower layer of the epidermis or upper layer of the dermis. So thereby avoiding the interaction of needle tip with that of the pain receptors. So thereby we will not have the pain. So this is what is seen in case of this particular figure. So then why microneedles? As we know, transdermal drug delivery is having certain advantages over the other drug delivery systems. But particularly the microneedles will be able to avoid the first pass metabolism of drug happened in GI tract as well as in the liver. And it also allows the rapid penetration of the drug into the systemic circulation because it bypassing the barrier layer. And most importantly, the needle phobia associated with the hypodermic needle can be totally avoided. Where you see in a picture, a boy, he is scared of the needle. So we can provide microneedle can provide painless and irritation pre-drug delivery. Then it is able to load the drug in the required amount, and the accurate dose of the drug can be maintained. Better drug stability. If you look at the vaccines are generally lyophilized and then it is kept in the vials. But if we'll talk about microneedles, we can encapsulate the vaccine in the microneedle and making them stable. And we can also provide controlled drug delivery by using biodegradable polymer in the microneedle preparations. And most importantly, last one is not the least, self-administratable. The patient can administer itself, uh, can administer it without the support or help of health professionals. So, but there are limitations, just like other techniques, it is also having limitations. But these limitations can be minimized. So, requirement of sophisticated fabrication processes will require. The stratum corneum thickness may vary depending on the part of the skin. Then less amount of the drug sometimes can be administered with the help of coated microneedles that can be avoided by using other microneedle techniques. And sometimes if silicon is used to manufacture the microneedles, if any part of the silicon or any part of the glass or metal left in the skin will cause irritations and inflammations. And cost of the technology is a little bit high, but as the time goes on, it will be also affordable. So what are the desired characteristics we need to make the microneedle technique a successful one? The dimension should be within this range, because if the length is more than this, it may touch the pain receptors. Then base, width, tip diameter, all those must be considered when you are manufacturing the microneedles. Then ruggedness, the insertion force. This is a major area where most of the researchers are doing, how to make the microneedles which will be easily inserted into the desired depth in the skin without being fractured. So that also you have to look at. Then penetration part, it must be or it should be able to deliver the drug in that required depth of the skin, because we can use the microneedles for intradermal drug delivery as well as transdermal drug delivery. Then reversible, whatever the damage or the pores, micropores formed in the skin must be healed or filled in less time. Then drug delivery, it is able to deliver the drug in both sustained manner as well as in the bolus manner. And margin of safety is also important. The margin of safety will be calculated by taking ratio between the force required for piercing the stratum corneum to the force at which microneedle breaks. Always less than 1 is preferable. Then we come into the next part, types of microneedles. There are many types of microneedles. As in the historical part, we have seen there are 4 microneedles we have talked about. Now another 2 microneedles came into the picture. The 5th one is hydrogel based, and more advancement happened in rapidly separating microneedles. We'll talk about each one. First is solid microneedles. As the name indicates, solid microneedles, it must be prepared from the material which can provide the sufficient mechanical strength, like silicon, metal, and sometimes polymers also can be considered. But the polymer must not be biodegradable or non-biodegradable. Here, solid microneedles deliver the drag in 2 steps. First is the pre-treatment of the skin by the microneedle, where the micropores will be created, called as microporation. Then the microneedle will be taken out, leaving behind the micropores in the skin. Immediately, next step will be immediate application of your formulations. It may be a patch or it may be a liquid formulation. If it is a patch, drug-loaded patch, the technique is called as poke and patch method, or the approach is called as poke and patch. They insert, then put the patch. Or if liquid formulation, first you have to create the micropores, then you have to put the liquid formulations which will flow through the micropores to the desired depth of the skin. Then we talk about coated microneedles. Here the drug material will be coated on the microneedles, and here after the drug material coated on the microneedles, then it will be inserted on the skin, and after some time the drug will be released from its surface. In the 3rd step, we are going to remove it. The blank microneedle has to be taken out, leaving behind all the coated drug materials inside the micropores. So thus this approach is called as coat and poke method. So next is dissolving microneedles. This is most widely used nowadays, because the 3rd step, generally we are talking about removal of the microneedle, blank microneedle. In the previous methods were not carried out, or not required to be carried out. Make clear. So here the materials considered must be soluble one as well as biodegradable one, and the drug is encapsulated within the microneedles. Then those microneedles will be pressed on the skin so that the microneedles will pierce the stratum corneum and reach to the desired depth, and will release the drug by dissolving the microneedles. So this is that's what called as poke and release. This approach is called as poke and release. So here the drug can be released in a faster manner as well as controlled manner. This is what makes this dissolving microneedles more widely accepted. Then we talk about hollow microneedles. Hello, am I audible? Hello. Hello. Hello, Gautam sir. Hello. Hello. Hello, anybody is there, sir? Hello. Hello, Kana sir. Are you there? Hello. 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 Am I audible? Hello. Okay, thank you. Thank you for the response. So next is hollow microneedles. Next category is hollow microneedles. Here the microneedles will be inserted into the skin, then followed by the application of the liquid formulations over the microneedles. And those liquid formulations will move through the pores of the microneedles and deliver the drug in the desired depth of the skin. Then after that, the microneedles, the black microneedles, the hollow microneedles has to be removed from the skin surface. Then the next category, the new category came is hydrogel-based microneedles. This is an integrated approach consisting of cross-linked black needles and the solid base plate to which a reservoir patch is attached. After the insertion into the skin, the hydrogel microneedle will swell by absorbing the fluid, interstitial fluids from the surrounding cells into it, followed by movement of the drug from the patch through the swollen hydrogel into the surrounding cells. So it is considered as poke and release type of your microneedles. Then this is a new advanced one, rapidly separating microneedles. Here, the drug encapsulated microneedles will be covered with a solid microneedles which will help in the insertion process into the skin. After the successful insertions, the outside solid microneedles has to be separated. That means it has to be removed, leaving behind the water soluble microneedles in the skin. This is also called as poke and release method. So there are a lot of your materials generally used to manufacture the microneedles. This is a big list. So metals are used, particularly for solid, coated, and hollow microneedles manufacturing. Glass, borosilicate glass is used for hollow microneedles, particularly due to fracture problems. Nowadays, it is absolute not used in the manufacturing. Ceramics are also used for solid microneedles. Polysaccharides like sugars, maltose, trehalose, fructose, raffinose, these are used for the manufacturing of solid as well as dissolving as well as hydrogel forming microneedles. Biodegradable polymers are also used. Non-biodegradable polymers are also used. Biodegradable like PLA, lactic acid, polyvinyl pyrrolidine, PLGA, and PGA. And non-biodegradable polymers such as alginic acid, PVA, polyvinyl acetate are generally used. So there are many manufacturing techniques to fabricate the microneedles. These are the few important manufacturing techniques. The first one is micro electromechanical systems. The use of micro electromechanical system to develop the microneedles. Here we see the 4 steps. First is deposition of photoresist layer. Before to that, the oxide layer is created on the substrate like silicon by applying high temperature around 900 degrees Celsius. After that, the patterning will be done with the help of UV radiation, wavelength in between 190 to 240. In this process, the mask will be used to filter the UV radiation and fall onto the particular part on the photoresist layer. Then etching means removal of the parts where the UV radiation fall. So, but here 2 things, one negative, one positive. Positive one, where the UV radiation fall will be removed with the help of etching solution. And negative one, UV radiation falls on that part and make it stronger by polymerization method. So the other part will be removed with the etching solution. Then finally, the photoresist layer is removed to get the microneedles. This is called as micro electromechanical systems. Then laser cutting method is used. It is a very widely used method where the laser will be focused on a particular part of the metal. Then that part will be cut to make the microneedles. Any type like in-plane microneedles, out-plane micro. In-plane means the microneedles in parallel to that of the metal. Out-of-plane microneedles means the microneedles are protruding from the surface of the metal or base plate. The laser ablation method, a laser will be used to create the plasma, that is electron and ion. That plasma consisting of ions and electrons will make the needle of this particular shape. Then micromolding method, it is also called as solvent casting method. It is widely used in our lab also. We can prepare this one. We have tried out this particular micromolding method where the formulation in the liquid form has to be poured onto the mold. Then it has to be degassed, removal of gas by applying the vacuum, then drying, followed by removal from the mold. Is the easiest way to develop the microneedles. The next method is atomized spraying method. Here atomization technique will be used to fill the micro mold, followed by drying and getting the microneedles. Then droplet borne air blowing method. Here simple droplets will be put on a platform or a plate, followed by another plate putting on the droplet. Then pulling the upper plate so that the droplet will be elongated. At that time, it has to be dried with hot air, and finally the plates will be separated to get the microneedles in both the plates. Then this came 3D technique. As we know, 3D technique is very popular nowadays. It is having few steps like the use of CAD, computer design, to first configure the structure. Then it has to be converted into the language, printer language, like STL file, followed by the STL file will be used to make 2D structures, two-dimensional structures by slicing. Then layer by layer, the structure will be developed by using the liquid formulations or solid formulations or powders. So fused deposition modeling is one of the 3D printing method where the liquid formulations will be dropped on the platform layer by layer to make the microneedles. Then in the coating method, coating of microneedles, we are having 4 techniques to coat the drug on the solid microneedles. So first one is gas jet drying method, where the atomization of the liquid formulation, liquid means where the drug and a particular solvent will be there, will be converted into fine droplets and dropped onto the microneedles to make a coat. Same way as a spray drying also being carried out. EDHA method is also used, electrohydrodynamic atomization process. Here the first drug solution will be passed through a channel, then a high voltage will be given so that the potential or the charge developed in the liquid gradually increases, followed by the passing of the liquid with a great force through the atomizer. And finally, the inkjet printing. Inkjet printing is again a 3D technique where the ink will be, that is the drug solution will be passed through a nozzle with the help of piezoelectric transduction, which will vibrate to create the droplets, fine droplets, and finally it will be coated in the microneedles. So after that, after the manufacturing, we have to look at evaluation part. Here evaluation is more or less similar. We can say many evaluation parameters are generally not carried out for the microneedles. What we have seen for tablet capsules, all those. Generally, these evaluations is divided into 6 categories or 6 your steps. First one is morphology. Second one is mechanical strength measurement, because that is what we need to insert or to create the insertion pores. Then skin penetration capability you have to measure. Then drug permeation, in vitro drug release. Finally, in vivo studies. So morphology case, what we generally measure is the dimensions, whether it is within the dimensions or not. Mainly the tip radius. The tip radius is very important in penetrating the skin. What methods or techniques we can use is same stereo microscopy. Then mechanical characterization, here we see mechanical strength and fracture strength to find out the safety consideration, where mechanical strength is carried out with the help of texture analyzer or motorized force measurement test stand, where having a gauge, the gauge will be moving up and down depending on the microneedle size. Then fracture test is carried out with the same or else confocal laser microscope. Skin penetration will be measured with the help of methylene blue staining process, where methylene blue will be used in the microneedle manufacturing and will be inserted into the skin. After the insertion, the methylene blue will be released. So by dissolving microneedle, there give a better picture about what depth the microneedles are penetrated. Insertion force displacement test. It is generally done on a hard surface to find out how much force is required to penetrate to a certain distance in the surface. Then insertion depth, failure force, and skin barrier integrity. This is also very important, skin barrier integrity. This is done with the transepidermal water loss method using a vapometer. It will give the indication whether the micropores formed with the help of microneedles are sealed or not. Then drug permeation. Drug permeation will be carried out with the help of French diffusion cell. And any skin sample we can consider. And what are the parameters we are going to measure are flux, permeability coefficient, and enhancement ratio. These three parameters are generally measured here. In vitro drug release. In vitro drug release case, we have to use your glass or beaker to put the microneedle in that and take out the samples for the measurement of drug concentrations. Then in vivo studies and the animals we have to do to see the pharmacokinetic and pharmacodynamic parameters. Then we look into the application of microneedles. This is very important. So here, drug delivery, vaccine and gene delivery, diagnostic, cosmetic and cosmeceuticals. So basically, vaccine and gene delivery, the mechanism how the vaccine is successfully delivered into the desired part of the skin is given in the flow chart, where after the vaccination by microneedle, the vaccines as an antigen will be taken up by dendritic cell and Langerhans cells present in the skin, which are generally called immunocells in the skin, but are densely present in dermis and epidermis, but not in the subcutaneous and muscle layers. So that is the reason why if we administer the vaccine with the help of microneedles, it is much more beneficial compared to that of your hypodermic needle method, where the hypodermic needle will bypass the number of dendritic and Langerhans cells, because those are the cells which will receive the vaccines and they will process it and they will present it to the T lymphocytes in lymphoid organs. Then it will evoke the strong immune response. So that's why the vaccine must be given with the help of microneedles, not only to avoid the needle phobia, but also minimize the loss of vaccine. Am I clear? So this is what a vaccine delivery. Drug delivery, as we know, the large molecules, small molecules, controlled drug delivery, fast drug release, all are possible with the help of microneedle. Then diagnostic purpose, it is nowadays widely used for diagnostic purpose as well. Am I clear? For suppose we talk about you're taking out of the blood samples, or else directly measuring the samples inside the skin, So there are methods we are using. The mechanisms are negative pressure method, capillary effect method, swelling method. These are the mechanism by which we can take out the samples. Transdermal, the next part is transdermal detection, how we can do. One is on-device diagnosis method, off-device diagnosis method. On-device diagnosis method is by inserting the microneedle, which is having a sensor, will give the reading about the particular your ingredients in the interstitial fluid or in the blood. Off-device diagnosis, or otherwise called as phlebotomy, where the blood samples will be taken out or any other interstitial fluid will be taken out from the skin for the outside measurement. Then cosmetics and cosmeceuticals, where it is being used to deliver the cosmeceuticals as well as to stimulate the natural healing of the skin. How the natural healing is done? By microneedle applications, which trigger wound repair mechanism, the inflammation, which lead to the production of collagen and elastin, which are the your fibers in the skin, maintaining the skin structure. Then filling of the tissues and thickening of the skin, so thus improving the skin appearance and firmness, smoothness when there is a disease conditions like scares, like acne, and other cases like scares, stretch marks, wrinkles. So there are microneedles which are in the market. So first one is in the market is derma roller. It is not only used for cosmetic purpose but also used for delivering the drug. In this, your derma roller, first the micropores will be created. It is considered as a poke and patch or poke and flow method. Microplugs. This is also another product developed by Josano Pharma, where the microneedles will be having the drug or encapsulated the drug will be placed over the actuator or the applicator. Then it is to be pressed on the skin surface to successfully penetrate the microneedles into it. Then Solvia, this is another one having a microneedle connected to the syringe. Is a hollow microneedle type. The syringe will be having the formulation will be injected. Then others are microstructure transdermal patches. Here we can see the microneedles which will be able to deliver the liquid formulations of various viscosities. Then microcore, it is dissolving microneedle type, having the applicator. Microhyla is another one. It is generally used for skin, your skin, your repairing part. Then we are having derma pen. Is a device actually used to deliver the formulations into the skin because it is having the microneedles on the tip. And finally, micron jet. This is where we are having a syringe connected to the microneedles, which is made up of silicon. Generally, 3 microneedles are there in this. So this is next part is patient compliance and safety considerations. This is most important because patient compliance will make any drug delivery system successful. Skin recovery process. It depends on whether you are applying and leaving or you are making the skin occlusive. If it is occlusive, the sealing process, the natural sealing process will take time, around 1 day or 2 days sometimes. So skin irritation and infections, where if biological incompatible materials will be there, then will lead to the skin irritation infections. Suppose I will tell the stainless steel microneedles. If it is a rusted one, obviously will create problem, infections and irritations. If any part of the microneedle is left over in the skin, if any non-biodegradable polymer left over in the skin, will cause a problem. And majorly, we have to look at the sterility. After the manufacturing of microneedles, it must be sterilized. Otherwise, it will lead to more infections. Then pain. It was found that below 550 micrometer length of the microneedles will not produce any type of pain. Then our research and where actually we stand. We published this research paper where we have used solid microneedles. We have used dermal roller on the skin surface, rat skin surface, to create the micropores, then applied the formulations. So we have compared the result obtained from microneedle to that of the penetration enhances, that is Cineol. Then we have checked about the synergistic effect, if exists, if we combine the penetration enhances with the microneedle. Then we found this result where if we use this only penetration enhances, the enhancement ratio compared to the control is 1.9. But if we use the microneedle, we have found that the enhancement ratio is 2.8, where the drug is ibuprofen. And then we have checked about the synergistic effect. We found the enhancement ratio of 3.3. So we have seen there is an increase in drug penetration compared to the penetration enhances. At the same time, we also found there is a synergistic effect on the drug penetration through the skin, where microneedles will able to deliver much amount of the drug directly into the dermis or lower epidermis. At the same time, Cineol, which work on the penetration enhancement mechanism of lipid solubilization in your stratum corneum. So both combinedly enhances the drug penetration, that is ibuprofen. Then where we stand, actually we have now prepared microneedles, dissolving microneedles we are preparing, developing with the help of your PDMS micromold with a novel technique that I will not mention here. So finally, the conclusion part. The microneedle technique process is targeted to reach the US $1.2 billion by 2030, with the compound annual growth rate is about 6.6. And due to that, we've seen lot of research on the microneedle, thousands of scientific papers, patents, near 2000. Many products are in clinical trials. Many are completed the clinical trials. Many are in the market. So now another field in the microneedle case is the applicator. Much more research is going on the applicator part, because if we use the microneedle with the help of thumb or any our fingers, the pressure may not be sufficient or may not be in the proper direction we are applying it, so may create problem. So that's what applicator is must in most of the cases. So how can we develop a better applicator to deliver the drug with the help of microneedle? That is our feature perspective. With this, I have completed my presentation. Thank you all for patient listening. And I thank AICT for giving me this opportunity. I thank the organizers, the Roland Institute of Pharmaceutical Sciences. Thank you. Any questions? Any questions, sir?
Dear delegates, if you have any query,
Yeah, I think there is a question. How we will sterilize microneedles? This is a good question actually. The microneedles will be sterilized. Generally, we have two methods. One is before the manufacturing of microneedles, another is after manufacturing the microneedles. In case of microneedles, it is prepared to sterilize after the manufacturing. Generally, the radiation method is used to sterilize the microneedles after manufacturing. And another part also we can talk about the ingredients. Whatever the ingredients we are considering, it must be sterilized one. And in the sterilized environment, it should be manufactured. Because anyway, we are going to insert the microneedles into the skin. There is a chance that any microbes may enter into the bloodstream. So the sterility must be done with the help of a post-manufacturing, that is by radiation method. Any other questions?
Dr. Ravi, I have a question. Can you hear me? Gautam sir, unmute yourself. Unmute. Sir, unmute yourself.
hello. So, Dr. Padhi, I have one doubt exactly regarding different types of microneedles. Which one is more effective?
Yes.
Yes, Gautam sir.
Actually, if you look at all these microneedle types, the dissolving microneedle is the best one. Why? Because we have to select actually the biodegradable polymers. We need not to go for the last step, that is removal of the microneedle, because the total microneedle is going to be dissolved out in the skin. And second, most importantly, the drug, how much drug actually encapsulated inside the dissolving microneedle, 100% is going to be delivered. So that is where dissolving microneedle is the best.
Can we prepare this in the laboratories? So
Yes,
what exactly
that is
we
what I am
need
talking
to
about, this
prepare?
solvent casting method, micro molding. But for that we need a micro mold. Micro mold we need. So micro mold can be obtained from various sources. As I obtained one, that is a silicon micro your mold from the Blue Care Technology UK. From the UK I
How much
obtained
the
that.
cost?
So
How
that
much the
type of,
cost?
yes, cost is one micro mold around 10,000 1 year back. So we can purchase suppose a different length in the micro needle, starting from 300 micrometer to 1000. That is the range generally we use.
Okay, another question is there.
Yes, uh, dear Rohit, yes, we can deliver the nanoparticles, because when we develop the micro needles, that time we have to encapsulate the nanoparticles in the micro needles, because nano size easily can be encapsulated in the micro size. So another point also, if we are having a micro needle arrays, arrays means number of micro needles will be there, and there will be a backing layer, which is generally we are calling reservoir. In that we can encapsulate the micro your nanoparticles, so that will be delivered slowly into the skin through the micro needles. So the patch is generally considered if we want to deliver more amount of your drug, because micro needles may not encapsulate sufficient amount of drug in it. So if you are having a patch behind it, it will be able to encapsulate much more amount of the drug.
Another question is there. You just go through this.
Yeah, which type of micro needle? So generally hollow micro needles can be taken. This is a good question. So hollow micro needles are generally used. Having the micro needle at the tip, behind that there will be a syringe. There will be a syringe in that. Am I clear? So that
And
will
another
helps in delivering it.
another question is also there by because the sir. Sir is asking
Yeah.
what about the stability of micro needles?
So stability of micro needle, it is not a problem for the micro needles, because whatever the drug material we are going to encapsulate inside, generally in a molecular form they will be present. So as such, there will be no problem in the stability issues for the drug inside the micro needle. But for as a common thing for other your drug delivery systems or dosage forms, we have to keep them at the required temperature and humidity. So another point I want to make here, suppose microneedle vaccines are generally encapsulated in the microneedle. To avoid the stability issues arises when it is packed in the vials and ampoules, because many vaccines are generally packed in vials in the powder form. It has to be reconstituted again. But in the your microneedle, if you encapsulate, so that problem won't arise. The desired dose will be there in the microneedle.
Okay, there is no more questions. So now request our coordinator sir to complete the.
Am I
Yes sir. Yes
audible?
sir.
Sir,
Sir, for accepting our invitation, and we are seeking your cooperation in future also. Your cooperation in future seminars, whatever we will be conducting.
Yeah, sir. Definitely, sir. Definitely. Thank you for this opportunity, sir. And
And please accept this
we can also.
certificate on
Yes, sir.
behalf of Roland Institute. Yes,
Yeah,
please go
yeah.
ahead.
Thank you, sir. Thank you, sir. Thank you, sir.
So please accept this certificate of appreciation on behalf of Roland Institute.
Yes, sir. Thank you, sir. Okay, sir.
1 or 2 minutes,
Yes, sir.
Dr. Ravi, so that you can interact with him also. Dr. Suryakant Sai,
Yeah. Good afternoon, sir.
sir,
Hello.
welcome
Hello. Good afternoon.
to the seminar. Dr. Suryakant Sai, can you hear me? Can hear you.
Yeah,
Your
yeah,
friend
yeah,
Dr. Ravi
yeah, yeah. Yes, my voice audible to you, sir?
for
Yes,
you
sir.
as the
Yeah.
first
It is really a nice
speaker.
presentation by Dr. Ravi Narayan Padi. I listened his entire session. I really appreciate his innovative session. And congratulations, Dr. Ravi Narayan Padi, sir, for motivating to our young researcher in the field of microneedles and its current applications. Thank you very much, sir.
Thank you, Surya sir, and all the best for the
Thank
presentation.
you.
So I
Okay, sir.
request now our honorable Dr. Gautam Kumar Jena, the co-coordinator for this program, to
I can leave, sir.
Okay, thank you, Ravi sir.
I can leave the studio, sir.
Pari sir, thank you very much. Sir, you can leave.
Can I leave, sir?
Yes, Dr. Ravi
Yeah.
Pari, you
Yes,
can
Thank
yes,
you,
leave
sir.
sir.
the studio.
Yes, sir, you can
Okay.
leave.
Thank you, sir. Thank you, sir.
Yes. So I request Gautam sir to introduce the next speaker, Dr. Suryakant Swai, to our participants.
Thank you, sir. Good
So the
afternoon.
Gautam sir, excuse me, sir.
Yes, sir.
Just now, principal sir also joined.
Okay, okay, sir.
Sir, principal sir, can you hear me, sir?
Yeah, yeah, yeah. Surikant, how are you?
Sir,
Namaskar, sir. What about you, sir?
I'm fine, fine. Thank you. Uh, Thank you for accepting our
Welcome, sir.
invitation and coming forward to
It's my pleasure.
share
It's my
your
pleasure,
knowledge.
sir. Yeah, it's my pleasure,
Where are
sir.
you now? Thank you. Thank
At Assam only, at office only, sir. At office.
you. Okay. Okay. Thank you once again. Niranjan, shall we start?
Yes, sir.
Well,
yes, sir. So I request Gautam sir to introduce our speaker.
Okay, sir. Thank you, sir. Very good afternoon to all delegates. And the speaker of the session, Dr. Suryakant Swai. I, Dr. Gautam Kumar Jana, Associate Professor, Roland Institute of Pharmaceutical Sciences, feel privileged to introduce to the second session of third day, slot 2 AICT sponsored STTP, AICT sponsored STTP and organized by Roland Institute of Pharmaceutical Sciences. Dr. Swai is presently working as professor and associate dean, School of Health Science under Assam Kaziranga University. Professor Swai has completed his PhD from Berhampur University, M.Pharm from BPUT, Odisha, and B.Pharm from Roland Institute of Pharmaceutical Sciences. He qualified in GATE 2006 Professor Swai has published more than 100 research and review articles in the Journal of National and International Repute. He has H index of 17 and I-10 index of 30. He has edited 6 national textbooks as per the latest syllabus of PCI for B Farm and M Farm courses and 35 national book chapters. Dr. Swai has received one Indian patent grant and published 2 Indian patents. He is presently guiding 10 PhD research scholars and already guided 60 M Farm and 40 B Farm and 7 Farm D projects. Dr. Swai has also received several awards such as ML Khurana Memorial Award, Rajni Bhai V Patel Farmanova Competition Award, etc. Dr. Swai has attended more than 100 national and international seminars and conferences. With this brief introduction, I welcome Dr. Swai to address the gatherings.
Thank you, Dr. Jena, Gautam Kumar Jena, Associate Professor of Roland Institute of Pharmaceutical Sciences, for giving my nice introduction. Always people are giving resource person speakers, they are giving acknowledgement at the end, but my acknowledgement is always beginning. So I am very fortunate that I am the product of Roland. I am a student of Roland. I am an alumni of Roland. I am a PhD scholar of Roland. I am a teacher of Roland. I am a researcher of Roland. What I have learned, that is from Roland. My career started from Roland. So my heartfelt gratitude, my all the teachers, staffs who guided me, who supported me, who developed my career. Really, I point out few of the teachers during my education at Roland. Before start my presentation, Dr. VVB Ravi Kumar, currently heading the principal position at Roland Institute of Pharmaceutical Sciences. Vice Principal Dr. C.H. Niranjan Patro sir as my MPharm guide, and my research guide, Professor M.E. Bhanuji Rao sir. Really, I am very thankful to them. And lastly, without motivation and without support of such a motivated management I ever seen, the chairperson, Madam Jayalakshmi Madam, Director Dr. J. Surya Rao, and Secretary and other trustee members, Dr. Sunil Kota, sir, my friend Dr. J. Shruti, and other teaching fraternity of Roland Institute of Pharmaceutical Sciences. A very good afternoon to each and every persons who have joined directly and indirectly in my session. Dear delegates, we know that we are passing a very crucial time in our life. So God, Jesus, Allah, they are also watching what we are doing during this situation. So based on this, first I am praying to God and to start my presentation. I really thankful to the AICT for giving me this opportunity. Under the AICT AQIS online short-term training program coordinator, Dr. C.H. Niranjan Patro sir, for his invitation, and other Dr. Gautam Kumar Jena and Kanucharan Panigrai sir. Thank you very much. Before I start my presentation, why I have selected this topic, because it is very relevant to the theme of this STTP program, that is integrated research in basic science uh, with applied pharmaceutical science. Dear friends, dear delegates, you know, nowadays quality is the major concern and major part. Not only academic teaching, research, any field or industry or any sector, quality is the major aspect and major tool. Everybody should know what actually the quality by design. No doubt, few of the lectures, few of the session, few of the conferences you might be attended previously, but still due to the stipulated time, I will give the fundamental concept related to quality by design concept to pharmaceutical application with special overview related to PLG loaded nanoparticles prepared or manufactured by double emulsions method. Dear friends, pharmaceutical quality by design is not a buzzword, you know. From the inception by ICH, Indian Conference of Harmonization, and US FDA, the concept of QBD has gained significant attention by the global biopharmaceutical industries. If you very specific to pharmaceutical industry, QBD has played important role for the product development with regards to efficacy and potential product. So the key behind QBD implementation was improving the manufacturing standard as well as to improve the quality of the product to get the desired goal, desired objective in the form of a robust and quality products with high therapeutic performance. So that's why quality is playing major role in pharmaceutical industry. But if you'll say as per the ICH Q8 guidance, the concept of QVT and its systematic science and risk-based product development using predefined objectives really minimizes the current challenges and the issues related to the quality, robustness, as well as the product performance. So that's why quality plays an important role in pharmaceutical industries. In the year 1990, the scientist J.M. Joran, he is the first person given the statement of QBD, not QBD, that is quality. So he always saying that the product should be quality product. If the product is not a quality product, it will having lot of regulatory issues. To overcome such regulatory issues, then you have to apply the QBD concept or quality by design concept with the different principle of optimization, principle of design of experiment, or the principle of design space, control strategies, and other elements of QBDs. So that's why quality is playing a prospective and implementing the quality practices in the pharmaceutical product development. So that's why quality is not by chance. Quality is by proper planning with proper objectives. So let us, if we'll go to my first slide, according to Janet Woodcock in the year 2013, Director, Center for Drug Evaluation and Research, FDA, what she said, a maximally efficient, agile, flexible pharmaceutical manufacturing sector that reliably produces high quality drug product without extensive regulatory oversight. I told you, quality not by chance, quality by proper planning, proper execution, proper objective, and with proper planning. Without planning, maybe there is a chance of failure of your product, whether it is a biopharmaceutical company or pharmaceutical company or any manufacturing company. Basically, the companies or manufacturing industry who are producing commercial product or exhibit batches. So that's why, according to Woodcock, what she said, you have to give the high quality drug product from your manufacturing industries without giving any regulatory issues after filing into the either INDA or for the approval of US FDA. Dear friends, drug delivery system, we are applying, we are discussing about the QVD concept in pharmaceutical drug delivery system. So in order to balance any drug delivery system, irrespective the specific drug delivery system, always we are selecting bunch of chemicals, materials, and we are adapting different techniques, different processes, different infrastructures, manpowers, materials, and environment. So these are the few targeted product profiles which is giving the desired profile for developing a suitable drug delivery system. So the drug delivery systems usually a intricative device, that means a very very complicated device. In order to get a suitable drug delivery system, we have to select a proper drug based on their characteristics, physicochemical characteristics. And prior to the formulation, we have to go for the pre-formulation studies. And after that, whatever the excipients available in the market, based on your target profile, based on your desired characteristics, based on your objectives, based on your delivery system, you have to select certain excipients, certain release controlling polymers, surfactants, lipids, emulsions, suspending agents, processes, etc. You see the plethora of excipient, it is a plethora means bunch combination, something combination of polymer we are using in some of the delivery system. It is not that much easy to screen out such variables, such polymer variables in your formulation design or preliminary formulations development of any drug delivery system. Then you have to balance your drug delivery system in a proper way in order to get the drug delivery performance in its own characteristics manner. So every day, every moment when we are basically involving in our laboratories, in our research lab, or in our FR&D department, what we have to satisfy our boss, satisfy our HOD, satisfy our research mentor or advisors. So it is not that much easy to take the optimal decision immediately for the satisfactions about your boss when you are engaged any project for the development of pharmaceutical product in your industry or in research lab. So that's why what you have to do every day, everyone comfort different situations, has diverse duties to perform, different persons to please with dissimilar needs, demands, and taste, has made options to choose forms. Dear friends, I told you you can't fulfill 100% your efficiencies in any sector, but you try to do it, you try to implicate it, so there is a chance to develop, or you can satisfy your boss. But how can you do all these things? So that's why you always select the environment which is fit to you, and you have to select which is relevant parameters, so that you can achieve your desired goal, and you can plan accordingly from the very beginning in a balanced, happy, and contented manner, so that you can satisfy your boss, but by choosing a few vitals among the different biased parameters. You have to discard, or you have to remove certain aids, failures, aim and objective, and very specific objective is required in order to optimize, in order to develop the product with a rational goal. So that's why the concept of optimization is comes into the field. So optimization is the key to keep things in the balanced and orderly manner. Dear friends, dear delegates, I told you in order to balanced in any sector, you have to prove it, and you have to show your talent. You have to believe the persons. You have to believe to your management, believe to your environment, believe to your surrounding, believe to your research team. Without believing, you can't develop a product which is showing quality attributes and quality product in a balanced and orderly manner. So to optimize such product and such delivery system, the design of experiment or the DOE tools is highly essential. To apply such tools, number of free tools, paid tools available in Google, or you have to subscribe from the licensing authorities in order to apply in your different delivery system or in your different research area, irrespective to the pharmaceutics. You may apply in chemical science, or in chemistry, or in basic science, or in pharmacological science, or in biotechnologies, any field, based on your variables and based on your responses, based on your quality target product profile, based on your QTTP, based on your CPPP, that is critical process parameter, based on your critical formulations attributes, critical material attributes. So these things I'll tell you later slide, but these are the things is required before applications of any software. You have to fix your level, and you have to find out in your preliminary trial patches what are the real variables affecting the desired response changes. So that's why DOE is one of the important software tools, design of experiment tools. You have to apply rationales so that you can find out the minimum trials from the number of preliminary trials, and you have to minimize the time, and you'll get the robust product, and you can improve the efficiency or efficacy of the product with the limited studies. So that's why the basic principle of DOE is to laying out the details plan of structured experiments based on appropriate statistical criteria in advance to performing the experiments. So before implementing any DOE software, or if you want to know something about the QVDs, if you want to apply something about quality by design or formulations by design or design of experiment tools, first of all, you have to know the fundamental concept, principle, basic principle of DOE optimizations and other related elements of QVDs. Then only you can apply in a systematic way, and you can get the validated products by knowing the different part of the DOEs. So first you have to define your objective, and you have to measure the plan of your objectives, and you have to analyze the DOE as per the input variables in the software. You have to improvise, and finally you have to control by finding the different responses curves that will be practically, it is not possible now, but I can show you few of the examples. Right. So different scientists across the globe, they have given different concepts about the optimization. I told you it is not very specific definition for one person. It is varies from persons to persons according to their applicability, according to their outputs, according to their product performance, according to the clinically it is proved with less regulatory issues. So systematic optimization using experimental design is a time and cost efficient assuring high formulations qualities. I told you, friends, that time is the major concern or major issue wherever you will develop your product. If you are extending your research, if you are extending your product development till the market, commercial market is simultaneously any of the manufacturing companies developing the quality product, then what should be the your boss reaction or your vice president reactions in industry, or your principal or your research guide remarks towards you. So that's why always you have to apply such applicability tools in your research so that you can get the efficient and assuring your product is showing some standard. Similarly, other scientists they have given, like Bhupinder Singh et al. In the year 2008, he is one of the renowned persons related to quality by design, and with so many scholars like Dr. Beg and other, Prashant. So these are the persons basically in India. They're applying most of the cases DOE concept, quality by designs concept, in order to get globally to prove that your drug delivery systems having some pharmaceutical application. And other scientists similarly define the objective of or the definitions of optimizations. So what are the actually the merits? There are number of merits, number of advantages about the design of experiment techniques, but I will highlight a few of the design of experiment advantages. Like it requires fewer experiments to achieve an optimum formulation. If you apply the DOE concept or quality by concept or FBD, formulations by designs concept, or by using software, then you can achieve your optimum formulation instead of doing in heat and trial methods for several days or maybe period of 2 months, 3 months. And you can find out the best formulations among the others by as per your objectives, and you can trace out and rectify what are the problems you are facing during the preliminary trials, and easily you can sort out this. And mass thing is that wasting of time and efforts, materials and cost you can minimize, and you can improve your resources, and you can generate the revenues with your research project, and you can predict the performance of formulations even without preparing them. And flexibility of experiment you can achieve by these applications of DOE concept. Yeah. So I already told you what is QVD. So QVD, first, that is the scientist Jaran, and no need to explain again. But dear friends, I told you when you will apply in your lab or in small scale industries, the productions may be very very limitations without transferring your product into the technology transfer. In best way I can tell you, when I did my research at Roland Institute of Pharmaceutical Sciences, during my research PhD time, whatever the product I develop, and based on the adaptations of that process, I got the patent grant, and now I initiated my product. What the grant I got from the Patent Office, Government of India, transferred to the Glenmark companies, initiated the filing according to the patent trademarks issues. So that's why always you believe that if your product having some quality, then you can transfer your product to the through the technology transfer into the productions area or productions companies or manufacturing companies with QVD concept, so that it will be commercially available with limited design space concept. So what is the new in it? KVD by design. So KVD is a culture encompassing quality principle and strong compliance to it. So I told you why we are applying the QVDs, because the major attentions and resources on what that is important towards our customers, towards our health professionals, for our patients, and always doctors or healthcare professionals, nurses, they always prescribing or suggesting to the patient the branded products with having limited adverse drug reactions or side effects. So in order to achieve such things, so in the preliminary process design steps, or in the screening stage, or before clinical trials, you have to develop your product with the applications of QVDs. So the major objectives of QVD is quality excellence, customer satisfactions, and productions without having any problems. So best price with best quality, you can sell your products. Apart from that, if you'll see, time is major concerns. You can minimize the money, resources, efforts, and you can share your knowledge, because I know something about QVDs I'm sharing now to you peoples. So you will also applied in the future, and you can improve time to market and consumers generic skepticisms when the consumer or patient need. So according to their requirement, you can develop the generic products with the applications of QVDs, and reduce the cost associated with poor qualities, and improve the minimize the post approval changes. This is very very important. Sometimes we are not screening out the design space or control strategies and all the elements of QVDs. So that's why whatever the products we are developing through the applications of QVDs, once it is goes into the commercial product or into the market, what will happen? So that will be a great issues related to regulatory point of views. So you have to again withdraw your product from the market, and you have to sort out where is the problem and what should be the measures you have to take for the secondary applications of QVDs. So in order to achieve the desired goal from the very beginning, then you have to set your goals, set your models, set your analysis in a proper way, and you characterize and analysis all the issues related to the QDS, and you have to shorten the lead times, and you minimize the requirement of materials and resources, and you have to screen out the edge failures related to your product, and recall the fewer products which increase the profit margin of your products. Dear friends, optimizations in case of formulations development, if you are applying QVD concept or optimization using the statistical designs tools, then you can say formulations by designs with the approach of optimization. So before applications, at least you have to know, so 5 steps of optimization of formulations designs. First is, what is your formulations objective? When you will start your research, whether it is a PG or UG or your PhD synopsis, first, what is your aim and objective, or what is your rationality behind selecting such topic? Second thing is that once you have chosen your formulation related topic or delivery related topic, then how can you screen out your influential input and response variable or dependent and independent variables? So selecting such variable and screening such variable is not easy without performing the preliminary trials or the number of batches. And by varying the different variable concentrations, percentage, or the percentage of seed, you may not finding the exact influential variables or responses variables related to your delivery systems. Then you have to go for the formulations development with the applications of as for the design of experiment with the different types of design of experiment, I'll tell you. And then you have to evaluate the analysis of variances, analysis of by applying the statistical analysis ANOVA, and analyze the different curves, different plots, and statistical fit, fit, p values, level of significance. So many things is there in the softwares. According to that, you have to evaluate it. Then modelizations, you have to find out which kind of model it is fitting to your designs, whether it is a quadratic model, polynomial model. So different types of model according to your inputs, according to your response, according to your level coded values. And next year, the last step is the study validation, scale off, and productions. So the key to formulations by design is a strategy to set off experiments such that the information required is obtained as efficiently and precisely as possible. And verily, it's a statistical designs defining size and number of experimental unit, manner of their allocations, and appropriate type and grouping. That means in very simple way, which type of design you want to apply, where you should apply, and why you should apply, and what are the labels. Everything you have to know, then only you can apply the experimental units with the proper allocations, with proper designing, proper grouping. Without knowing statistical designs tutorials or tutorial for experimental designs, or whatever the designs is available, whether it is a screening designs or response surface design, you have to know all the tutorials from the very beginning, then only you have to apply. So few of the design I can tell you: screening designs and response surface designs. Screening designs, initially you have to screen out your influential response variable with related to the independent variables. So you have to screen out among the different types of or selected variable which is the major influential variables among the others by applying the screening designs, various type of screening designs like Tagosi design, like Plackett-Borman design. So like that, these are the examples of formulations by design, by screening designs like optimals, fractional factorial designs, Tagosi design, PBD, that is Plackett-Borman design, and response surface design like FD and CCDs, BBD, Box-Benkham design, and simple mixture design, D-optimal design, I-optimal design, fractional design, so many other designs also there. It is not specific to, because these are the examples, some of the experimental design which is commonly or mostly applicable in case of formulation. So the major part I want to discuss this today session, that is design space, because dear friends, design space is the important part of element of QVD's quality by designs concept. Without knowing the design space, you might be not applicable in the QVD concept. So you see here, in any design space, again it is having four different space, like explorable space, knowledge space, design space, and control space. So what actually these all the spaces? It is the multi-dimensional combinations and interactions of material attributes and process parameters which provides assurance of the qualities. When you will do your study, your formulations design within the design space level, then definitely you can control and you can find out, you can screen out your last product or last formulation batch among the several trials or several batches. So within the design space, you can decide, or you can control the optimum product, optimum batch, and the remaining whatever the formulation you have prepared during this knowledge space or explorable space, this is not at all considerable during your formulations batches. So movement within the design space is not a change. That means if you want to change your level from here to here within this area, that is acceptable. But once you will change your level, suppose this is the maximum high level of the concentration of X and the concentrations of Y, but within this level you have to set off your goal, set off your coded values, and you don't go to the knowledge space. Maybe you are thinking that why not, suppose 50 gram of HPMC is showing better release retardation of drug from the control is product. But the reality is that after increasing the concentrations of HPMC, what is the release profile? If you go to the studies, in some cases 100%, 100 something percentage, or maybe decreasing, increasing. So that indicate it may be a variable, it is not under your controls. And explorable space means it is completely failure batches, so you never consider these batches. I'll tell you in the later slide what actually you see. When you can find boundaries around the critical process parameter, or whatever the critical process parameter, your QVD element or critical material attributes or critical formulations attributes, you always find out after preparing the suitable trials of formulation what are the boundary of batch failures. Because you see 1 2 3 4 5 6, all are, this is, here is the question marks, is all the formulations batches really it is a failure batches, because it is showing is the edge. But you have to screen out this edge failure. Evaluation is very very important according to the concept of design space. You don't think that all the batches are failure, but sometime you have to change and you have to control the critical process parameter, you have to control the critical material attributes, you have to control the critical formulations attributes, and so many things. So in order to evaluate the edge failures, so what are the real edge failures and what are not the edge failures, and what is the important of design space. So we want to management the quality risks, and design space can illustrate the understanding of the parameter of interactions among the independent variables. Design space can include critical and non-critical parameters. Design space should validate the operationals at full scales. But you see, out of six, what actually the real, sorry, the real failures, these three are the real failures, but these things we are considering because it is under the control space, it is under the design space, but it is really, it is not the edge failure results it is showing. So after evaluation you have to confirm it. But here the question is how you evaluate this evaluations process by applications of design of experiment tools. So that's why always we have to consider the control space area or the design space areas by which we can develop our product. And after doing the several, characterization studies, whether it is a drug release studies or by entrapment efficiency or friability, hardness, whatever the other parameter related to, different delivery systems, we can find out the exact design space locations. If I'll say, here two parameter you have selected, parameter 1 and parameter 1. Parameter means what? The concentration of something, concentration of your variables, independent variables, right? So how You see here, this red area is the point of failure. If you are getting such type of graph during the evaluation of edge failure in your software, then it is indicated is the edge failures, and this green area is the point of success. And the more success this square area design space, here you will get the maximum success rate, no doubt. And this is the area by which it is giving the informations to you that you can prepare within this level your formulations. And this important of the design space is always, product requirement can change as per the market needs and federal compliance, resulting the different optimum operating the design space. Whenever the market need, no, we want the DISO of 99%, not the 90%, then again you have to expand the design space, and you will go for edge failure evaluations and proper design space options, depictions, or predictions. And sometimes the expansions and the contractions of the design space is having some questions why we should expand a design space, when is design space expansion possible. If you go to the different cases when the original design space was artificially constraints for simplicities in case A, case B when some edges of the design space are the same as edge of the knowledge space. So that means, yeah, I told you there is number of edge failures, but again you are screening out from the edge failure after evaluation that these two are not showing different results according to our desired objectives. But after selections, again you have to expand it. For example, you are selecting two of the response variables, temperatures, and no, not response variable, independent variables, that is temperature and seed weight. Then what should be the design space area, and what actually this showing? This showing, see, if you'll find out in the second case two, the same parameter, same responses, you are selecting temperature and this, or out of this area is located, red area is the edge failure, this is the area, this is the success. But what actually, this is the low level, this is the high level, and within the level of temperature from, let us, 10 degree to 60 degree you are changing, and with the variations from low level to seed percentage from this to this, what is the success rate, and where is the design space, where is the control space, and how should we predict it? So the predictions is always giving the software, and based on the margins, based on the design space concept, you can predict what is your success rate. So if you'll see in this case, this large square represent the ranges test in the formulations designs, and this is the area represent the point of failures, and the green area is represent the point of success, and the more success within this area. And that's why, what is the major variance related to the design space? Here, three variants, 1, 2, and 3. In the first case, this is a normal operating range. Dear friends, this is the complete design space area, including the control space. Within the number 1 and number 2 variant, what this is the normal operating range. You can do whenever you want, you can prepare this, final batch optimized product. Suppose today you develop this product, it is already available in the tomorrow market. After 1 year also, you can utilize this formulation composition, and you can develop the same product and send it to the commercial market. But in this case of design space concentrations, optimized concentration of your base, optimized base, there is no regulatory submission. No need to submit any regulatory issues raised by the FDA or US FDAs. But if you are beyond this design space, beyond this space variants, whatever the product you are developing, that is abnormal operating range. So there is a regulatory submissions required. You have to submit the regulatory issues raised by the US FDA or the authority, European agencies, wherever you are applying for your approval of your products. So this abnormal operating range, always you have to discard, and always you have to select this 1 and 2 variants, because there is no regulatory submission. And this is the design space which is operating the entire internal chains. Whatever you want, you can change it. This is under your knowledge, and it is not under your beyond your knowledge. So if you'll say the regulatory importance of design space, this is 3 types. One is the lab scale design space, pilot scale design space, and commercial scale design space. commercially is always acceptable if you are applying the design space, but if you will go for lab scale design space and pilot scale design space, because there is no regulatory importance, we are not sending our product to the commercial market. But when you will send to the commercial market, then and there is a regulatory concern, then you have to fulfill as per the FDA guidelines or ICH guidelines. So that's why proper knowledge from the very beginning and with the end in the mind is very, very important when you want to develop any pharmaceutical product from exhibit batch to commercializations. So development studies prior to that, knowledge is required, product understanding about your product, about your formulations, about your delivery system, and process understanding, which method you are adopting and why you are adopting, what is the risks related to the process, related to the product, related to the materials, related to the batches, and what type of sometimes if QVD is giving some kind of errors, then you have to apply the PATE, process analytical tools, which can minimize the quality associated problems or quality QVD related errors. And after that, you can enhance your product or process by your experience, manufacturing operations experience with the concept of design space. Then gradually you can improve your product with less regulatory problems. You can change within the design space, and there is a continuous verifications by the US FDA regularly about your product if it is available in the market. So this is about a few of the computers related tools which is available in licensed versions, and few of them are free versions, trial versions. You can apply in your formulations or in other part of your research according to your domain areas. That is computer use in systematic optimizations, like for your delivery systems. And these are Design Export, Statistica, JMP, Miniture, Start Graphics, Umetrics, and Multi-Simplex G, Spectop, SAS, Optima, JMP Minitive, Design Export, Trial Versions, Designs Ace, Name Drops, Scoped, SCAD, GRG2, ADD. So these are the few software available in licensed versions. Either it is annually subscriptions or lifetime subscriptions. But the one important thing is that in case of formulations design, it is not easy to apply the statistical tools which is available right now. But for that, what is your main delivery system, you have to select it according to your delivery system, according to your if you are preparing a sustained release triplet, if you are preparing a microsphere, if you are preparing nanoparticles, vesicular drug delivery system, macroparticulate system, fast releasing tablet, self-emulsifying system, or any other delivery system. It is varies from delivery system to delivery system applications of different kind of softwares. You see, this is the most of the times experimental tools or quality by design or formulations by designs right now available in the market literature. If you find out, this is the area number percentage of sustained release per tablet is prepared so far, developed in the market by the applications of QVD, similarly in other delivery systems. But if you'll see the market literature values about the different software applications available, marketed products with the concept of design space or control strategies, that is formulations by design, BBD, Boxman designs, central composite designs, de-optimal designs, that is simplex lattice designs, extremes vortex design, so many other screening design, or which is right now fractional vectorial designs is applicable in market more, and it is available in commercially most of the places. And if you'll see up to 2010, sorry, I have not updated up to 2021 because this is my previous informations. Apologize for that. No doubt, if you'll see up to 2010, between the oral and non-oral delivery system, if you see the oral delivery system have significant applications of QVD concept compared to non-oral delivery system. If you'll see one of the literatures as per the critical review therapeutic drug delivery system journals, which is published by B. Ising et al., that is the number of response and number of independent variables related to the literature reviews about the formulation designs concept. The variations of literature is showing that number of response variable and number of independent variables. Response variables with the literature report is, initially it is very high and later stage it is decreasing. But when you are comparing the literature report with the number of independent variables related to the formulations irrespective of the delivery system, as gradually people are not considering the one of the independent variables which is required in the early stage in the year 1990, when the concept of QBD is derived by the J.M. Joran. But in the later stage, people applied and both the variables, and they got the suitable results or quality results. For the systematic optimization of drug delivery system, how you have to select this number of response variables and the independent variables, it is depends on the delivery system. Suppose if you are preparing microparticulate system, what are the process variable you have to select, and what are the response variable you have to select, and what kind of design of experiment or design you have to select or choice. Basically, depending upon the nature of mixing in the blood and the number of non-filled continents, steering rate, time, duration of cross-linking process, and injections rate during the manufacturing operations. And you can go for selection of response variables like in vitro drug release, entrapment efficiency, percentage yield, and you select out of this choice of design. You can select any one. Similarly, vesicular delivery systems like independent variables, you can go for that selection of contents of phospholipid and other lipids what you are using in your delivery system during the formulations trials. And the hydrations, medium volumes, time, temperatures, sonications time, frequencies, response variables like percentage drug content, drug loading, dermal retentions, release profile, size, leakage, and stability is related responses. Similarly, other delivery system, osmotic drug delivery system, polymer content, orifice size, coating levels. And these are the few of the examples of independent and dependent variables, and the choice of design what you want to use. If you are preparing biodesive system, then you can select type and amount of biodesive polymer, concentrations of rate controlling polymers like eudragids or any HPMC derivatives, and what are the response variable: biodesive strength, swelling index, drug release, percentage drug content, entrapment efficiencies. And most of the time of the literature is showing that fractional design or factorial design is a suitable design for that. You can apply for biodesive or microadhesive or gastroretentive drug delivery system. So if you are preparing a lead delivery system which for enhancing the viability of drugs, then you can apply for example SNADs, self nano-emulsifying drug delivery system, or nano emulsifying drug delivery system, or solid or liquid nano emulsifying drug delivery system. Then you can see what is your phospholipid, what are the surfactant, co-surfactant, co-mixtures you have used in your manufacturing initial operations, and what kind of response you have to select. These are the few, that is emulsification time, chemical stability, dissolution profile related variables, and zeta potentials, and polydispersibility index. So many things is there when you will do the characterization, preliminary characterizations about your formulations. Like that, other delivery systems and the choices of designs and response variables like microsphere, vesicular systems. And after applying all the designs, you'll get this type of curve in 3D and 2D plots. If you are getting this is the maximum plot, this is the minimum response plot, and this is the saddle response plot, this is the stationary reads, this is the rising reads. So this type of plot definitely you will get when you will apply the QBD tools or design experiment tools, and according to their shape you have to say what is the variations related to your changing in the independent variables about your response, whether the response having greater impact on changing on your independent variables or not. And according to their 2D plot and 3D response plots, you can analyze it and you have to interpret accordingly. For example, in case one, that is critical quality attributes, and one is the critical quality attribute parameter, and two, let us, we have selected the critical formulations attributes. So parameter 1 and parameter 2 are the critical formulations attributes, and the dissolution is the critical quality attributes. So dear friends, dear delegates, critical quality attribute is nothing but your response variables, and critical formulations attributes, your material, what materials you are taking and what is the variations about your materials, about your polymer or excipient you have used during your product development. So parameter 1 and parameter 2, and this is the 2D plot and this is the 3D plots, and you can analyze it how it is helpful if you are changing the parameter 1 from low level 40 to 60, and similarly from 0 to 2 of parameter 2, and what is the response about the dissolutions, percentage of drug release or percentage of drug dissolves. So this indicates, parameter 1, there is initially the 80% of the drug, and from when the concentrations of parameter 1, the percentage is 40, and gradually it is increased then followed by decrease. So here there is increase the concentrations of the parameter 2, what are the excipient you used, and that is influentially increasing the response of the dissolutions. But you can get the better idea if you see the 2D graph. You see this graph, this point is indicated is the control space, or this is the this area is entirely the control space, and or you can say this is the design space, and this design space control the control space, and this is your explorable space where you can get most of the edge value related to your parameter 1 and parameter 2. If we combine in that 2 CQA and 2 CFA, that means 2 critical quality attributes, 2 responses and 2 variables, independent variables. Let us, you have taken parameter 1 independent variables, parameter 2, and your one of the quality attribute, that is dissolution, and the second quality attribute is the friabilities, and this is the friability design space, this is the dissolution design space suggested by the software. And if you will find out the both the responses with according to the variations about the independent of 2 parameters, and what is your overlay plot is showing. This overlay plot is showing that is the combined design space for the both the critical quality attributes of friability as well as the dissolutions. And within these responses, you can prepare your optimized bets, and you can I analyzed, and you suggest that this is my optimized base according to the combined design space or overlay plot based on the selected response. If we go for 4 CQA, that is 4 critical quality attributes, or 2 critical formulations attributes, 4 responses and 2 independent variables. Let us 2 independent variable in case of self-emulsifying drug delivery system, that is 1 excipient parameter 1 competitors and 2 natives, and the response is particle size. Second is release 20% of drug release, and with a specific time point with the 2 different concentrations of independent variables. And the third one is entrapment efficiency, and fourth one is zeta potentials. If you want to find out the combined design space of 4 critical quality attributes with the 2 selected critical formulations attributes, then this you'll get, this is the area of the total design space area where you can find out the optimum results based on the overlay plots. And this overlay plot will give the particle size, optimum particle size based on the overlay plots, and entrapment with 85%, and the release with this, zeta potential with this, with the variations of x and y with independent variables, like selecting x this polymer and this excipient, or 280, whatever you have selected or what you want to take during the operations. And for example, if you will say one case 1, we are selecting time versus percentage of drug release. According to your target, you have to find out your optimum formulation base from the response tables, from the experimental design table. How you will select? Let us, our objective is within 9.11 hour, I want to know the 60% drug should release. whether it is a control release or whatever the release you want to, and basically it is applicable for control release because 9.11, not for immediate release. If it is your desired objective, and according to the desired objective, whatever the suggested trials suggested by according to your experimental design, if this is your feasibility search for optimum formulation space according to the experimental design with the different coded values from -1 of parameter 1 to +1, that is the low level, and this is the high level, and this is the low level, and this is the high level, and these are the time points. You see, these are the time points you got from the feasibility search table from the experimental tools or the software, and you have to find out where is the 9.11. So if you'll see in any other case, there is no 9.11. According to the setted levels from the very preliminary trials, if you get this 9.11, the time, then you have to mark it. This is the selections for formulations having the desired profile. Okay, desired profile, and that 9.11, and what is the level of parameter 1 and parameter 2. So what it indicates, the level is -1 for concentration of polymer X, and the +1 high level of polymer Y. If you will choose these two variables and you prepare according to your method of applications, definitely you'll get your desired formulations objectives. So I told you my special focus on PLGA concept based nanoparticles. That dear friends, PLGA nanoparticles can improve the therapeutic efficacy basically for gene deliveries due to sustained release and control release of the encapsulated DNA for over a period of times, not for 1 month, it may be several weeks compared to other polymer-based nanoparticles. So the PLGA, that is, poly D lactic co-glycolic derivatives, biodegradable polymer, played important roles basically in gene delivery or cancer targeting for developing the nanoparticles by the applications of QVD approach using the double emulsions manufacturing operations by selecting different quality by designs elements like CTP, critical process parameter, critical quality attributes, critical material attributes, with the applications of QVD in the pharmaceutical unit operations time. Whatever the design you are applying, whether it is a factorial design or box Mancom design, you will get the lipid solid nanoparticles, or maybe the nanoparticles with the combinations of the initial PLGA based with your selected drugs with the adoptions of the double emulsions method. But dear friend, the selections of qualitative target drug profile and critical quality attributes for the PLGA-based nanosuspension nanoparticles using double emulsions method is not easy. So first you have to fix your target and what is your quality attributes, and you have to describe all the quality attributes related to the dose of your drug, route of administrations, dosage forms, particle size, about your nanoparticle, polydispersibility index, zeta potentials, assay. In case of, I am speaking about this PLGA nanoparticles with applications of double emulsions methods, and what are the pharmacokinetics and oral viability about your quality attributes, about the impurities of the drug, microbial assay, stability, percentage of yield, and what are the residual solvents, and critical attributes like quality attribute like particle size, about the nanoparticles, polydispersibility index, zeta potentials, assay, drug release, and so many other parameters you have to select. And when you are selecting all these quality attributes, then you have to fix your objectives. What actually you want? What is the assay you want? 90 to 110%. I want zeta potential plus minus 30 millivolt. I need the particle size between 100 to 300 nanoforms, and I want to deliver this nanoparticle in a oral route, not a non-oral routes. And you want to fix that improvement of viabilities after preparations. So if you are fixing all this target, then according to the critical quality attributes, what are the risk assessment related to the critical material attributes and your inputs. This is very, very important. So if you are selecting all these critical quality attributes, let us, you have selected particle size, polydispersibility index, entrapment, drug release, drug product. These are the parameter, and you are changing this initial RICS, API solubility in aqueous phase, type of organic solvents, PLGA polymer grades, and type of WYO emulsifier, and what are the type of stabilizer, because different type of stabilizer is there. According to the nature of the stabilizer, you have to select, and according to the nature of the emulsifier, you have to select, and according to the ratio of PLGA with derivative of lactic acid or glycolic acid, you have to select for the preparations of PLGA nanoparticles. But you have to follow certain steps and certain methods for preparations of PLGA based nanoparticles according to your selections of manufacturing operations, selections of polymers, selections of stabilizers, selections of surfactants, and determining the potential critical quality attributes, identifications of critical material attributes, and applications of design space within the limit for getting the product quality products. So these are the certain variables which you may apply during the preparation of PLG and nanoparticles with a different ratio with lactic acid and glycolic acid derivatives of the monomer of PLGs, and the degradation strength is according to the ratio. The degradation strength of PLG will varies, and the HLV value or best, and the HLV value also you have to select the stabilizers, common stabilizer for preparations of PLGA based nanoparticles. These are the so many nanoparticles best stabilizer available in the market, and their zeta potential value also you have to consider. And these are the requirement of double emulsions preparation of PLGA nanoparticles. Basically, internal aqueous phase, organic lipid phase, external aqueous phase. And these internal aqueous phase include your drug and the vehicle. And organic lipid phase include polymer and your emulsifier, and the solvent, organic solvent, what you have used, and what are the lipid you are selecting in and the polymers. And external phase is always include the stabilizer and based on the values, target values, based on your this HLV value, you have to select it. Next, vehicle also you have to use in the external aqueous phase. If you see the general method procedures for preparations of whether it is industry or in the laboratories in small scale preparations, first you have to take your drug in the inner aqueous phase and mix with this polymer or lipid in the outer oil phase, and then after combinations you will get the product in the form of emulsion, that is WYO emulsions. Then additions of primary emulsions into this aqueous phase with constant steering with a high speed, then with a controlling speed, or that means high speed means with controlled speed with the homogenizers with the addition of suitable concentration or optimum concentration of stabilizer, you'll get the secondary emulsions in the form of WYO by W, and then you can recover by removing the solvent. And this is the general scheme for preparations of PLG nanoparticles by double emulsions methods. And these are the few CMA and CFA and the critical process parameter. Basically, you have to select when you want to prepare this critical development of PLG based nanoparticles. If you see the Fishbone diagram or Ishakya diagram for QBD perspectives for the pharmaceutical applications of double emulsions for preparations of PLG and nanoparticles, when any manufacturing industry or in R&D, there is two sides. One side is your manpower, your environment, and your resources. In the second stage is that what are the things you need for preparation for a suitable delivery system. Like that, if you'll see the quantity of organic solvent what you are using, and organic to aqueous ratios, API solubility, type of organic solvent, PLGA grade, PLGA viscosity, polymer to drug ratios. In the second part of the your critical quality attributes or critical quality attributes for preparation of nano suspension, you have to select the stabilizer type, steering or homogenization or sonication speed, and the HLV value for the selected stabilizer, solubility of the stabilizer in aqueous phase, and the method what you are applying. So that's why for maintaining the life cycle of any quality product for better understanding and the management of the life cycle of QBD based PLG and nanoparticles. So this schematic representations is required. First is you have to understand about the product what you want to prepare, and you have specific knowledge about the PLG and nanoparticulate delivery system, and what kind of CQA, CMA, CPS, CPPS is used, basically selected for preparation of such type of delivery system. And then you have to analyze the process and apply the process, and you can understand the process. You have to control the strategy about the design space and perform the process. And the last, I want to conclude my session. That is, applications of QVD tools in the development of pharmaceutical product is considered is the best approach to meet the product quality for the patient benefit. And QVD is omnipresent in the entire product development life cycle, and can be considered as a versatile tool for attending desired safety, efficacy of the drug product to meet the consumer needs. I told you in very simple way, only we are applying the QVD concept for benefit of the patient, and to give the quality product into the market. And the complexity of the formulations and minimization of the complexity of the formulations, removing the different error associated products, show that QVD plays an crucial role in the manufacturing sector during the product development, for maintaining the quality of the life or throughout the life cycles with lower or fewer rejections, and decreasing the cost about the pharmaceutical product, and approval process shorten based on the applications of QVDs. If you'll see a few of my recent publications in SN Applied Science, this is the recent acceptance letter what I have achieved, that is quality by design prospects pharmaceutical application in double emulsions method of PLG loaded nanoparticles, which is recently accepted. And these are the few other publications in our team, that is introduction to experimental design in springers. Few other related, specifically QBD related product with my teams. These are my profile of different publications, whether it is research, review, or chapters related to quality by design only. Okay. So these are my publications, and also book related to QBDs. And this is my patent with applications of design of experiment. This is another patent, and these are my achievements. And these are the few, literature references I have used for preparing this, all the slides. And at last, again, I want to acknowledge to the management of Ruland Institute of Pharmaceutical Sciences, Principal Dr. V.V. Ravi Kumar sir, Vice Principal Dr. C.H. Niranjan Patro, faculty members, professors, and other non-teaching staffs associated with the Ruland Institute of Pharmaceutical Sciences. My sincere thanks to my friend Dr. Gautam Kumar Jena for his motivations and encouragements. I extend my special thanks to AICT event in charge and other team members who have invited me. I really thankful at last to all the participants and delegates for patience of hearing my sessions. Thank you very much, and stay safe while staying your home. If you have any queries, you can raise your questions. Thank you very much.
Thank you, Dr. Suryakant Swai, for your elaborated presentation on quality by design, and at the end giving the real practical tips by showing some of the case studies related to PLGA and other formulations. We are really thankful to you for your for giving your valuable time and sharing your knowledge with all the participants, and we are expecting similar kind of cooperation from you even in future. Thank you, Dr. Swai.
Yeah, is welcome, sir. Thank you very much.
So the session is open for question answer. So I request the participants, if there is any question, please type it in the chat box. So I have a question, whether this
Yeah.
PLGA based formulations are suitable only for sustained release, or they are also suitable for some sort of targeting?
Yeah, definitely. It is applicable for both control is, sustained is, targeted delivery, because I told you PLGA polymer, improve the therapeutic efficacy and viability of the drugs, basically for gene delivery or vaccine delivery. That gene delivery, vaccine delivery, it is a targeted delivery system. So PLGA also applicable for targeted delivery system. And when you are targeting any delivery system, it is not for short time. It is maybe more than a week or several weeks. So it is applicable for all.
Okay, thank you.
Welcome, sir.
So, any more questions from the participants? Okay, thank you very much. So,
Welcome, sir.
on behalf of
Welcome, sir.
on behalf of Roland Institute of Pharmaceutical Sciences, we are presenting a small certificate. Please accept this certificate of appreciation from Roland Institute of Pharmaceutical Sciences.
Thank you very much for giving me this opportunity. Once again, thank you to management, principal and vice principal and other staff. Thank you very much.
So, there is a question from one of the participants. How a new
Yeah.
researcher will apply QPD guidelines in lab scale? How a new
Yeah.
researcher will apply QPD guidelines in lab scale?
Yeah, very good questions. I really like it. When a unknown persons, blind persons, they don't know about the activities, how he or she will apply during the laboratory research. I told you KBD is not specific to lab, not specific to industries. It is, commercially applicable to each, whether it is a exit base or in a lab scale or in a industrial scale. But before applications of QBD in your lab, first of all, you have to know the qualitative target product profiles or elements of QBD and what actually the QBD is, what is the concept of QBD, and how we want to apply the QBD for our formulations. Because really it is required the QBD. Without applications of QBD, I told you so many disadvantages. To minimize such demerits, then you have to apply in either laboratories or in industries. Basically, you have to find out your formulations variables, influential variables, and you have to screen out by the concept of QVD or by the concept of design of experiment tools.
Thank you very much, Dr. Swain. Actually, we are really
Welcome, sir.
proud that Roland Institute has got an alumni like you, who is making or
Thank you, sir.
working as a brand ambassador for the institute because of your knowledge, hard work, and I'm really happy to see your in-depth knowledge in the area of quality by design. Thank you very much, and I hope again in some other occasion you will be a speaker for our programs. Thank you very much.
Def- Definitely, sir. Thank you very much for constructing me such a teacher, such a students like you, and motivated teacher, sir. Thank you very much, Professor Dr. Chinam Niranjan Patra, sir. Really, I am grateful to you, sir, for your initial advice and suggestion.
Okay, thank you, Dr. Swain. So today's program is coming to an end, and my dear participants, please join tomorrow again by 11 o'clock. Thank you all.
Thank you,
Thank
everybody.
you, Dr.
Thank you,
Swain.
delegates. Thank you, participants, and Principal VVB Ravi Kumar, sir, and management. Thank you very much. Sir, I am ending my sessions. Thank you.
Yes, thank you.
Thank you, Dr. Jana.